Hello, I'm going to speak to you about protein phosphorylation on arginine residues, which is a non-canonical form of protein phosphorylation. There are several things that are special about it. For one, if you look here, the phosphoryl moiety is attached to a nitrogen rather than oxygen atom. This makes it chemically different and, for example, unstable at low pH, which in turn makes it difficult to detect with some standard laboratory techniques and mass spectrometry techniques. Also, the arginine side chain is positively charged, while the phosphoryl is negatively charged. And this unusual charge distribution is actually what the pockets that detect phospharginine, and we found 3 such pockets and characterized them structurally, uses to identify phospharginine. So it detects it with complementary surface charge distribution. So people often ask if this modification is found in human cells. Well, there are some indications from proteomics that it might be, but really the only kinase that is known to target arginine residues in proteins is McsB, and this is a bacterial protein. So that's the structure of McsB. When we characterize this protein structurally, we confirmed what could be seen on the sequence level also to some extent, that it is actually homologous to some animal proteins, including human proteins, but these are not protein kinases. So in the human cell, there is a protein called creatine kinase, which is a metabolic enzyme which phosphorylates creatine, which is a small molecule that has the same chemical group that arginine does, but it's not part of the protein chain. By lacking this lid domain here, which is present in creatine kinase, McsB is accessible to large protein substrates. Instead, McsB has this domain shown in yellow, which has 2 functions. It mediates dimerization, and also it has here on the side a phospharginine binding pocket. Now, because McsB autophosphorylates on an arginine residue on the other side, the McsB dimers, and this is illustrated here in this diagram, the McsB dimers can self-assemble into an octameric cage. And the pins here represent the phospharginine residues, and the holes are phospharginine binding pockets. So we also managed to crystallize this octameric cage. And Bence could show with in vitro phosphorylation assays that it has some preference for unfolded substrates that would be able to access the active sites that are buried inside the cage. Now, in terms of the biological function of this system, the main one seems to be to target proteins for degradation, which is illustrated here. So a bit like a ubiquitin signal, but in gram-positive bacteria. And the central point of this system is a phospharginine binding pocket, this time in ClIP-C, which is a part of a ClIP-CP proteolytic complex. By recognizing phospharginine, this protease specifically degrades phospharginine carrying proteins. And this can be reconstituted both in vitro and also shown in vivo, where the wild type cells would degrade some of the substrates, but kinase deficient cells wouldn't. So thank you for your attention. And here are some publications that describe these studies. And I thank all the colleagues from the Clausen group who contributed to these results. Thank you very much.